section. So then if someone for any reason missed the talk or want to see it again, it's going to be uploaded in our channel at YouTube. Please, I already see that most of you have already turned off the camera, which is great because then it doesn't get too heavy in the system, and also the microphone. And the questions in the end of the talk, please, and please make questions. It's important that we have your feedbacks. So what you can do in the end, it's either pressing the raise your hand button, which is, I believe, somewhere here. Mine is, I think for you, it's going to be on the bottom side. So you can check it. You can raise the hand, and I think it's going to be in the order. So like the first to press will be the first to make question. If you don't want to make question yourself, you can write it on the chat, and we can make the question for you. And if someone wants to write in Portuguese, it's fine. And we try to translate to Simone, okay?
I might be able to understand it.
Okay, so is it already another skill that you have now?
No, it's because of Italian.
Okay. Yeah, maybe if you know Spanish, it's even better because I think Spanish and Portuguese, sorry, Spanish and Italian are quite
more similar. Yeah.
Yes, but okay. So please make questions. It's important for us. Okay, just to show that this is the next talk. Simone also know this guy here, Fight. So later we are going to announce it. So this is the talk for next Wednesday and same time. Later we will put the link here. So about Simone. So Simone is going to give this talk. I know it's going to be very interesting. So he's assistant professor at the Albert Einstein College of Medicine. He's a really prominent, and I can say young scientist with more than 80 papers. And this is quite a lot considering his age. I'm not going to tell him that's he's like 25 years old. But, but he's not only good in science as well. He's like a really, really good musician. So he's like a very good drummer. This is like the B&B project that he used to have in Denmark when we met. So yeah, without further ado, I want to give the talk to Simone, and please, I'm going to now stop sharing my screen, and then you share yours as well, okay? And then, yeah, we try to make it work.
Yeah, thank you, Tiago. Let me see if I just do this. Oh, it's working, right? You can see my screen, right?
Yeah, sure, we do.
Perfect. Yeah, thank you, Tiago, for the invitation. Thank you, everybody, for being here. As you noticed, it doesn't seem to be a secret that Tiago and I, we go way back. So it's an emotional day for me. But you Brazilians are very emotional. I learned in America to hide all of my feelings. So I'll try to make this presentation interesting for you. It's not just about what we do in the lab. Since this is a lecture, I also wanted to show you something that maybe will help you think about mass spectrometry as additional ways to look at your samples, to look at your data. And that's why I call it 5-dimensional mass spectrometry, because beside the usual quantitative dimension, can we quantify proteins, for example? I think that there are a few other things that you can do with your mass spectrometer, and hopefully some of these are new for you. And we are very excited to show you what kind of technology we develop in our lab. First of all, let me tell you about what is our target. We study mostly chromatin biology, and chromatin is the control panel of every eukaryotic cell. Right? It's inside the nucleus. It's composed mostly of DNA, and as you can see in the picture on the left, the DNA is wrapped around other proteins, mostly histone proteins. You see here an X-ray picture of a nucleosome that is composed of 8 histone proteins. And then the view that we have about chromatin is this figure is beautifully simple in its way, because there are regions of chromatin that are highly compacted, inaccessible to transcription, and then there are others that are open, and then where there is gene expression, and this is how we think that cells regulate the expression of genes, and by consequence the phenotype, how they look like. All of these histones have post-translational modification. This red and green dot that you see, they benchmark regions of chromatin that have silencing modification and regions of chromatin that have modification that instead recruit the transcriptional machinery. When you Google histone modifications, you usually get tables like this. So there are 7 or 8 modifications on histones that they seem to be associated to gene expression. And then there are a few others that are associated to gene silencing, the green and the red ones. And unfortunately, the reality is much more complicated than that. And it makes sense because cells are very complicated. If you dig a little deeper, you will find that representations like this. And what you see there in that schematics, you see sequences of these histone proteins, and on the top, you see a lot of tiny writing, and these are all histone modifications that have been discovered with different kinds of techniques that decorate histone proteins with myriad of these post-translational modifications. So it's not just about active, inactive. There are so many histone marks, many of which we still don't know what they do. And this figure is old. This figure is back in 2014. I couldn't find a more recent one that gave the perspective. But just to give you an idea, since then, we have been discovering so many more histone modifications, including markers like serotonin and dopamine. And these modifications end up modifying histone proteins. So if you think about it, probably every protein modification that you know of has been found on histones as well. Plus, there are a few that so far we only know to be on histone proteins. And to make this even more complicated, it's very rare, and you will see it with our data, it's very rare to find on the chromatin histones that have only 1 or 2 modifications. Most of the time they are hyper-decorated. So you see that already the theory that one modification is active, the other one is repressing, is already not making sense anymore. And to make it even further complicated, is that we don't know the biological function of each of these modifications, and we know even less about what is the function of coexisting modification. There was a hypothesis raised up by Professor David Alice here in New York about 20 years ago about this histone code, where these histones, with all of these modifications, they work like keys. They fit into a lock, and the lock are these chromatin readers that process DNA readout. And 20 years later, we are still pretty far from proving if this is completely true. And part of the problem is that we don't have the technology to do that, because imagine developing an antibody to map each of these combinations. They are potentially infinite. So there's no way to raise an antibody for each of these combinatorial modifications. And to make it just one step more complex, and that's why we do this in a school of medicine, is this picture that you've seen. This represents a typical chromatin in a normal, healthy state. In reality, when during viral infection, while we age, in certain types of cancer cells, chromatin looks quite different. Chromatin gets unfolded, and then you have this three-dimensional state that also impacts gene expression. You see, for example, here you have silencing histone modifications that are inaccessible chromatin, and this is a cartoon showing that there can be gene transcription over there. So these domains are difficult to study because what is specifically in this domain, what kind of histone modifications we have, how they get there, and which of them are critical. If you have an active and a silencing mark, which is the one defining whether there is transcription or not. So these are our 5 dimensions. So what do we need to know to be able to characterize these domains and potentially identify, for example, a target for a therapy, or define whether this is a biological feature of aging chromatin? The first dimension is what we normally do with mass spectrometry. We can purify proteins, for example, that are associated to these domains with some kind of immunoprecipitation technique, and then study which proteins are there, how abundant they are, and what kind of histone modifications are there. But as you can see from the complexity, knowing which proteins are there might not be enough. So what we need to know, we also need to know, are there combinatorial modifications? Do they coexist? How rapidly they get there? They come from specific metabolism. Can we block this metabolism? And then how accessible are these chromatin domains, and is there transcription? And I'm going to show you now that some of these things can be done with other technologies, but I'm going to show you now how we do all of this just using a mass spectrometer. We need this toolbox, and then we need a model system. So in the last 10 minutes, I'm going to tell you how we model this kind of chromatin state in the lab using a cell culture hood. So first of all, mass spectrometry. I guess you guys probably already know about this, given the title of these lectures. But just a quick overview, you know about proteomics. Proteomics is where we extract proteins from the cell. We normally digest them into short peptides because they are simpler to analyze than intact protein. And that is this bottom-up workflow that you see up here. So and it works for histones too, of course. We digest histones into short peptides, and then we do liquid chromatography coupled to mass spec. This is a horrible figure, but that's my very first review, so that's why I like it. And when you analyze peptides like this, you see what is the problem, right? The problem is that you can quantify this modified histone peptide, but you frequently lose the information about whether they coexist on the same protein, because now there are 2 different molecules quantified independently. But the advantage of using this procedure is that it can become very quick and very robust, which is not a small issue. Let me give you just an example. When you run a histone peptide sample into a mass spectrometer, you run it with liquid chromatography. This is a 45-minute chromatography. You see, you can visualize this data in 3 dimensions, like the time when they come off the column, the mass to charge of these signals, and then the y-axis is the intensity of these peaks. And basically, you quantify these peaks like you quantify any peptide in proteomics. Histones, they have peculiar problems. For example, they have peptides that are called isobaric, where you can have a modification in one side or another. They end up having the exact same mass and generating the same chromatographic profile. So what we do, we have developed some software to be able to perform relative quantification based on the profile of their fragment ions. So I'm not going to get into details because it's boring, but you might want to consider this even if you do other kind of omics, like for phosphoproteomics. You can have peptides that they're just quantified together, but in reality they're very different species. And when the mixture is not too complex, for example, when we are purifying histones from a cell, we don't have a lot of proteins. We have a lot of modified peptides, but then it's not as complex as an entire protein. So what we can do, we can even consider doing some of these analyses extremely rapid without liquid chromatography. What you're seeing now in this video, you're seeing a device that we have in the lab. It's called the Adbion Nanomate. And what this robot is doing now is picking up a clean tip, is picking up your sample, and then it's going to spray directly the sample into the mass spectrometer. So now, remember that mass spectrometers, they scan at the rate of milliseconds. So they are never the bottleneck of the analysis. The bottleneck, the reason because an analysis takes an hour is because of liquid chromatography. So if you can bypass that, you can make very rapid injection, very rapid characterization of your sample. And then in this reference here, we show how we do this for a histone mixture. And we actually discovered that the main advantage is not just using a rapid analysis, but bypassing chromatography for a number of reasons. One of them is carryover, another one is inconstant performance, another one could be the difficulty of the software in always extracting the exact chromatographic profile. For all of these reasons, when you do direct injection, you reduce a lot the variability of your analysis. What this violin plot is showing you what kind of variation we get with normal chromatographic runs. With proteomic variation, usually they are around 20–30%. When you do direct injection, you go down to 4–5% because you don't have any more this problem, all of these problems that I just mentioned. So for specific application, we use direct injection to analyze histone peptide. You might think about it for some of your applications, you might consider it too. You might not have the nanomate, but you can do it with the series. The problem is about histones is that they are really complex. So what you see in this graph, you see instead how a chromatography looks when we do a middle down injection. When we do a middle down injection, it means that instead of cutting histones into short peptides, we cut large polypeptides. And this sequence specifically that you're seeing right there, this is the entire tail of histone H3 in human. And what I forgot to tell you before is that histone tails, so the part of the histone that protrudes outside the nucleosome, this is the part where you most likely have most of your post-translational modification because it's more accessible. And so analyzing the tail of histones doesn't give you the perspective about all the modification of the protein, but the vast majority. And in fact, if you look at this chromatogram, what you see up there, this is a helix chromatography. What you see up there, looking early, you see this hyper-modified histone modification. Where we quantify those peptides, we can tell how frequently modifications coexist. Now, the problem of isobaric forms here is, enunciated like 1,000 times, because you see all of these different forms. If you have phosphorylation in a different position, if you have a different combination of methylation, acetylation in different positions, these all have the same mass. So it's very complicated to do computational deconvolution of all of these species that are co-fragmented together. But you can check out some of our papers about how we do it, and you get at the end, maybe if the experiment works, usually it's a little more than 1,000 of these combinations, but there is a lot of biology you can do with that. And before I flip to the next slide, I would like you to notice one little detail. You remember I mentioned before that it's very rare that in a cell that you have histones that are only modified with a single modification. If you look at that chromatogram, look how intense are the peaks where histones are hyper-modified compared to how barely detectable are these peptides that do not carry any modification or just very few. And this is really showing you how unlikely it is to find histones with a single modification on your chromatin. That's why it's so important to look at combinatorial patterns. So when we analyze these long histone tails, not only we can tell something about which modifications coexist, but we can tell something about the histone variants as well, because there are histone H3, there are many types, at least in human. And then they actually differ of very tiny changes in the sequence. But when you look at this long polypeptide, you can tell them apart because, of course, they have a different sequence, and so they have a different mass. We can study how these combinatorial modifications, how frequently they coexist, and we can also use some computational normalization to tell how likely we find two histone modifications on the same protein. So why is this interesting? Because you can produce all of this data, but then you need to interpret them. So why is it interesting to know, for example, that two modifications are always associated on the same histone protein? because you can predict the function of some of them. And I'm going to show you just an example here, which is should be simple enough to be indicative. So this is an old study now. It comes from work that we did when we were still back in Denmark. And for some reason back then, nobody had done a characterization of all of the histone modifications on the embryos of C. elegans. So we were collaborating with a group that was working with C. elegans, and they really wanted to know the histone modification profile. So we did it, and then what you see there in that graph, this is the relative abundance of histone modification on the tail of histone H3. Of course, there are other histones as well, but H3 is a very good example. What you see here, you see the different residues, and then on the y-axis, you see how abundant are modifications on these residues. So those who are familiar with chromatin biology and histone marks, they immediately notice that something is really wrong here, because the very famous lysine 9 di and trimethylation that are typical silencing mark of human heterochromatin, they almost don't exist in C. elegans. And on the other hand, there is this column here with lysine 23 methylation, which is almost 50% of the entire chromatin. And these modifications don't exist in humans. We don't have lysine 23 methylation, or if we do, it's a very, very low abundance. So the question was, what does lysine 23 methylation do? And because we acquire our data using middle down and look at the entire histone tail, we could look at what are the most frequent co-frequencies of these histone modifications. And one example is the little lysine 9 trimethyl that we detected, that we know very well, that is a silencing mark, was always present together with this lysine 23 methylation. So because they are on the same histones, they cover the same chromatin domains, and because they cover the same chromatin domain, maybe they have the same function. And to keep a long story short, yes, lysine 23 methylation is a heterochromatic mark in C. elegans, and we generated the hypothesis by looking at coexisting modifications. Okay, now what if we find that post-translational modification is interesting for us? How do we follow up? How do we make this translational? So first of all, we need to find what proteins read those modifications, because maybe we can predict inhibitors that prevent this binding. And if it's typical of cancer, maybe we can, this inhibitor contributes to kill cancer cells. We should identify what are the enzymes that are writing, erasing those modifications. But something that is potentially simpler is how this modification gets there. So protein modifications, they come from metabolism, right? So acetylation comes from acetyl-CoA, methylation comes from methionine, phosphorylation from ATP, and so on. So when we do mass spectrometry, we can track how modifications get there, because we can feed cells with isotopically labeled metabolites. And then we can tell, is this the metabolite that generates this modification? And how long does it take once the cell eats it, or the animal, to become a histone modification? I'm going to give you an example that I hope you will like it. This is a work that came out sometimes last year. And that looks like a Nature cover, was not accepted because it came out with the same issue where Google presented the quantum computer, so of course they got the cover. But I thought the story was interesting. So what happened in this lab? They gave to these mice ethanol, alcohol, and this alcohol was labeled with deuterons. So instead of having this 3 hydrogen there with the asterisk, the alcohol molecule had the 3 deuterons group. And I'm sure you know very well that alcohol in your body gets converted into acetaldehyde, acetate, and acetate is a precursor of acetyl-CoA. So is this alcohol a precursor of histone acetylation? So the short answer is yes, but that was not the interesting part. The interesting part was what enzyme uses this alcohol to produce histone acetylation. So if you inject histone peptide into the mass spectrometer of these mice that are fed with this alcohol, you see what you see here is a spectrum with an isotopic pattern. And then you see that after 4 hours of, well, it's not 4 hours of constant drinking, but after 4 hours of the injection, what you see that the pattern of this acetylated histone peptide has changed. And that's because this isotope here is the same molecule that now is carrying acetylation that comes from this deuterated alcohol. You see, plus 1, 2, 3 Dalton, 1, 2, 3. This is exactly what you would expect. So by quantifying the intensity of this isotope, you can tell how much of this ethanol has been converted into histone acetylation. And to note, this spectrum specifically here is from the liver, but the same pattern you see it in the brain cells. And what the group was interested in this enzyme here called ACSS2, and it was asking, this enzyme is responsible to convert acetate into acetyl-CoA. But is this acetate coming from us drinking too much alcohol? So when you look at, when you extract the brain cells of this mouse and you look at the profile of this histone modification, you see that if you have the enzyme, this is the wild type ACSS2, this is the knockdown ACSS2, you see there is basically no difference. Like all the histone modification in this brain cell, they look the same. But when you look at how abundant is this isotope, you see that in the knockdown of this enzyme, this radar plot shows the intensity of the isotope, the +3 isotope. You see that this is completely gone, which means that, yes, the overall distribution of histone marks is not changing, but now we can track where that acetylation was coming from. And you know, that was pretty much the story. They were interested in this specific enzyme. So you see, we do this, again, we add this additional quantitative dimension by doing the same analysis in mass spectrometry. Okay, but then the most critical question for us is, are these modifications functional? So we find the histone acetylation, is it inaccessible chromatin? Is it promoting transcription? And this is something that is very difficult to answer with proteomic strategies. You do it with genomics. Right? With genomics, you can study what part of the DNA is accessible. You can study whether there is transcription. It's difficult to associate this with combinatorial modification because remember, we don't have antibodies to do chip sequencing. So how can we do that? We have two strategies in the lab that we now normally use for different experiments. And they are similar but somewhat complementary. And these are the fourth and the fifth dimension in a histone analysis. So one is looking whether histones and histone modifications are accessible on the chromatin. Well, you see, we extract the chromatin, we sonicate it. So the chromatin that is more accessible, when you break it with sonication, it creates smaller pieces. And then the inaccessible heterochromatin remains in big balls. So what happens is that you can physically separate those fractions, for example, by gel or by sucrose gradient. So you can analyze separately the small pieces of chromatin from the big ones. And then the small ones represent open chromatin, the big one represents inaccessible heterochromatin. And another one here on the right, we can feed the cells again with isotopically labeled metabolites. But now, instead of labeling modifications, we can label the protein sequence itself. You know, you probably have heard about Silac. This is exactly the same concept. If you grow a cell in a Silac media with this heavy labeled amino acids, when the cell produces new proteins, these proteins will be isotopically labeled. And why is it interesting for histones? Because all histones are actually synthesized in a specific stage of the cell cycle at the influence of the S phase, when the cell produces more DNA, and all canonical histones, I should say. But what is interesting is that the regions of accessible chromatin, where there is high transcription, this nucleosome has a faster turnover. So they get exchanged more frequently. And this is intuitive, right? Because you have the RNA polymerase passing through many times to transcribe your gene. When the RNA polymerase is passing through, it's kicking out the nucleosomes because it needs space to create a copy of RNA. So all of these nucleosomes, they are repeatedly kicked out from the chromatin, evicted and replaced. And then what happens is that if you label those histones, you can see that the histones that are in this actively transcribed chromatin, they have a higher labeling rate than those that are in the more inaccessible chromatin. And again, you quantify this with mass spectrometry. You can look at the heavy to light ratio and tell, is this modified histone in transcribed chromatin domains? So, so why is this interesting? Because when you combine the two data sets, you can see things that break some of the dogmas that we have today in chromatin biology, which is a specific histone modification does a specific function. Doesn't seem to be true. We know is more complicated than that. So look at this graph on the left. What you see here, you see a profile of how abundant is a peptide when it's quantified in the different fractions of this accessible chromatin fractionation. So the later fraction is the more inaccessible chromatin, and then you see that you see a higher signal down here, and that makes a lot of sense, right? Because we know that this histone modification, they are typical of condensed heterochromatin. We see those peptides in the late fractions. But if you look at histones that contain this heterochromatin marker plus an acetylation, then you see it coming out in a very early fraction, which means that yes, this histone is carrying a silencing mark, but it is accessible. It is in an accessible chromatin domain. So when you compare this with the other data set, of course, if you look at acetylation marks, you see that they have a higher labeling rate. So the histones that carry acetylations, they get a faster labeling incorporation, which means they are on histones that are faster, they have a faster turnover on the chromatin. Again, this indicates that there is a high transcription for the histones that are modified like this, on the chromatin domains where we have those histones. And again, this makes perfect sense. It makes perfect sense that the regions occupied by the silencing mark, there is no transcription because you have a very poor labeling rate. Now, what about the hybrid marks? The hybrid marks, they can have very interesting ambiguous behaviors. So this example specifically, what you see that a histone modified with this binary code, silencing active, is in accessible chromatin domains, is exposed to the outside, but there is a very poor turnover for the histones that have these binary marks. What is it suggesting? It's suggesting that histones modified like this are in open chromatin, but there is no transcription. Why that is, it's an answer we don't know yet, but what we're working on this combinatorial code, and this is, we think, is another step to unravel this histone code. The last problem is we need to model this in our cell lab because we are not rich enough to have all of these animal models, plus it's a lot of killing, plus all of these animal models, they're not human, right? So ultimately, we also want to have an overview about the human chromatin. And the other problem is, of course, that if we use human cells, we need to use them in a cell culture. And the problem of typical, let me say, cell cultures are extremely useful for a lot of projects. The problem is that the main goal of growing cells in cell culture is to keep them alive. And we can do that, but how do we keep them alive? We keep them alive because they constantly replicate. They constantly duplicate, so we always have fresh cells to perform experiments. Now this is not what happens in a solid tissue in your body. For example, if you take liver, you don't have liver cells duplicating every day. You don't even have liver cancer cells duplicating every day. So the problem of a cell constantly busy in replicating DNA is that it doesn't accumulate some of the histone markers that are typical of older cells, of adult tissues, solid tissues at least. So that's why we say they're not as functional for the type of studies that we want to perform. So we've been thinking about, we need to find something in between that is not perfect, but is a compromise between the two. So what we do three-dimensional cell culture. Three-dimensional cell culture per se include a very big family of different types of models and experiments. What brings them together is the fact that you accumulate cells on one another to form this little organoid or spheroid. There are many different ways of doing it. One way is to accumulate cells, for example, at the bottom of a well plate. And this is one example of the static system. It works. You can create those spheroids, but it becomes really hard to exchange media into this droplet. So in the end, you have to perform this experiment rapidly because these spheroids end up going into necrosis quickly. On the other hand, you have dynamic systems where you can constantly exchange the media and you can constantly give new food to this spheroid. But as you can see, some of them, they can be a little tricky to handle, not super suitable for high throughput experiments. So we found an interesting compromise. Interestingly, we found it back in Denmark with our old friend. What you see there in the video, you see Stephanie from our lab, who is working with the cell vivo system. So what she's holding now is holding the tablet that we use to control our bioreactors that are spinning into our incubator. And then what you see inside the incubator, you see our 3D cell model. So what these bioreactors do, they're in this constant rotation. So these balls that you see, these are spheroids of hundreds of thousands of cells that are packed together because instead of adhering to the bottom of the plate, they adhere on one another because they are in this no gravitational state. Actually, there is gravity, of course, because we are on Earth, but they are in this omni-gravitational state. So they are in constant spinning. So instead of falling to the bottom of the plate, they adhere on one another. And they create these big balls where there is a lot of cell-cell connections, cell-cell communication. It's starting to look a little more like a solid tissue. This is how we do it. We grow our cells for 1 day into this microwell plate. So we adhere the first 5 to 10,000 cells. Then we transfer them into this bioreactor. You see this very tiny dot. These are our cells that are starting to grow. And then when it spins like this for about 3 weeks, in the end, we have these balls that are more than 1 millimeter in diameter. And at this point, we have functional cells. And I'm going to show you data that prove that these cells are functional the way we want. It's a very smart bioreactor. You see that there is an opening here to exchange the media. You can exchange it with the syringe. There is an opening here to get this spheroid out. And what is interesting is that in these bioreactors, they're just filled with normal media. You can grow up to 300 spheroids. When we need one for proteomics, we just pick it up with our pipette, and then we process it for proteomic experiments. So we don't need to trypsinize, for example. We don't need to detach them from plate, which could create problems to cell signaling. So the first question you may wonder is that, are these alive? Because if these big balls inside, they have a lot of necrotic tissue, then we are looking at dead cells. We're not looking at functional cells. And data suggested that, yes, they are alive, and we try to prove this in many different ways because we are very concerned ourselves that we are just looking at dead cells. What is interesting is that these spheroids, after the first 3 weeks, they start growing at a much slower rate. So they go into senescence, and that's exactly what we want. Like, we don't want them in the constant proliferative state. When we break them and we look inside, we do tunnel staining, for example. There is no trace of necrotic markers. This is the positive control, how it looks when there are dead cells. And we also look at this using a different assay. It's called adenylate kinase. And yes, there are some cells that are dying. During development, you see this increase of this percentage of dead cells. But then they stabilize at a low percentage and is not this percentage is quite comparable with just growing cells in a flat culture on a Petri dish. This was done back by the developer in Denmark 7 years ago. We did this assay also into our lab, and we got pretty similar results. And this is very important because it's hard to have a reproducible biological system. And we managed to reproduce this 7 years of difference with different users, and of course in different continents. So this cells are alive, but the question is, do they look like liver cells? So specifically what we're growing here, we're growing a cell line called the HepG2C3A. This is an immortalized hepatocyte. So these are liver cells, basically. When they are grown in 3D, when you stain them for these different macromolecules, you can observe a different architecture. But what is potentially more interesting is that when you look at specific metabolites that are typical of liver, you see that after these 3 weeks of growing, they start to produce levels of ATP, urea, and cholesterol that are comparable to what you see in real livers. And this was very encouraging for us. Of course, we did proteomics as well. And then we took, again, the same cell line, we grow it in a 3D, or we grow it in a flat culture. And what you can see, for example, down here, this volcano plot represents protein abundance in 3D versus 2D. You see that the proteins that are typical of liver function, they are all enriched into this 3D culture. Liver produces albumin, even though albumin is mostly abundant in the blood. Liver produces proteins that have to detox you from metals. And then, of course, liver do a lot of metabolism, and 3D cells, they have much more cell-cell signaling. So that's what you see when you look at proteomic data. Now, specifically, we are interested in knowing if chromatin is more physiological, because that's what this is all about. Like, we need to model chromatin dynamics into our lab to be able to apply our technology. And chromatin is a challenging macromolecule, but we have some indication that suggests that we are going into the right direction. For example, chromatin histones, they have multiple post-translational modifications. One of them that I haven't told you yet is clipping. So adult cells, they also have histones where there are enzymes that they chop a piece of their tail. And these are still functional histones. They are still present in adult cells. You don't see them when you grow cells rapidly in cell culture because you are constantly producing new histones. So this phenomenon, this histone clipping, gets diluted away. You don't see it in your 2D cells. Even if you take exactly our cell model, if we grow it in 2D, there is no trace of this clip histone. You see it when you grow the cells in 3D. You see it in human liver. You see it when we grow our model, at least for a couple of weeks. You see that the clipping here is forming, and we can detect it. So there are phenomena that they take time to form in cells, and we can only detect them in our cells. They are, they stay alive for a long time without dividing. And another thing about non-dividing cells, so cells in a more of a senescent state, is that they produce larger heterochromatic domains. Like I told you, the new histones are synthesized during the cell cycle at this stage. And then if this cell is not cycling anymore, if it remains senescent, we produce more of these heterochromatic markers, specifically those on histone H3. There's a long story about these two markers. They are actually different from each other. You see from these 3D pictures that they form very different chromatin domains inside the cell nucleus. But this is not the topic of today. What is supposed to be interesting here is that, yes, senescent cells, they have increased amount of these heterochromatic marks. And if you look at our 3D cell culture growing, the more it grows, the more it looks like a senescent cell, where you have more of these diene-tri methylations on the lysine 9, which is a bit diluted away when cells are rapidly dividing. So this is what we're looking for. We got some confirmation that these cells are in a senescent state, because if we do metabolic labeling, if we feed these cells with heavy labeled amino acids, it takes longer time to produce heavy labeled proteins. In fact, you see the flat cells, after 1 day, 2 days, 3 days, 4 days, they already produce 30% of new proteins. And then when you grow cells as a spheroid, of course, this takes longer time, which is exactly what we expect from senescent cells, that they don't produce as much new proteins as a cell that is in constantly dividing. With this, I just have 2 slides and I'm done. The final and most critical question, and to be really honest, it's a question we haven't solved yet, so that's why we're working hard in the lab. Now we have these cells that can model chromatin state of adult tissues, and we have all of this technology. Can we model conditionally accessible heterochromatin? You remember the original figure where we have this cancer cell where the chromatin is all messed up? Can we actually reproduce that in a cell lab and look at this with our method? So this is a difficult question because modeling aging, for example, so where you have this chromatin decondensation, is really difficult. It's really difficult to model aging in a cell lab. So what we do, we use a couple of methods that give some effect, but we're still working on it. But I wanted to share it with you because I thought it could be interesting. One of them, we use chemicals. So if we use molecules like this, this one is a histone deacetylase inhibitor. So if you feed cells with this inhibitor, what happens is that the cells, they keep accumulating acetylation. And histone acetylation is associated with opening the chromatin. So you see that it's working day 1, 2, and 3. You have overall higher abundance of acetylation on histones. And then what you see these spheroids, they are starting to respond to this attack, to the fact that chromatin is aberrantly modified. Now, whether this chromatin is more accessible or not, that I still don't have the answer. We're working on this. And another approach, and then I'm really done, is we can take our spheroids, we can stick them in an X-ray machine, and we can induce DNA damage and oxidative stress. DNA damage and oxidative stress are typical indicators that this cell is old and the chromatin is old. And then when we do radiation on our spheroid, what we see that all the proteins that are responsible for UV protection, they get upregulated. So we know that these cells are responding to this radiation. We still have to do assays to show clearly that this DNA is oxidated. But of course, we did proteomics. And then when we do proteomics, we see a result that, to our eyes, they are very interesting. There are these proteins called high mobility groups, HMG, which are chromatin proteins. And these chromatin proteins are supposed to be upregulated, like higher expressed, when you have UV-induced DNA lesions. And we see that, but what is interesting is that we see it only when we put inside the X-ray machine flat cells. It seems like instead the spheroid cells do not respond to this radiation upregulating these HMG proteins. So we're a little concerned by this data. And then we have been digging deeply into the literature, and we noticed that somewhat the feeling is that this is true, but mostly in cell culture, which means that what we are modeling here might be something that is a little bit beyond the typical cell culture, because we give a three-dimensional structure to this tissue. And the question is, do we actually see the same response? And it seems the answer is no, which makes it interesting for us. But this is all still in progress, so I don't really have a final answer for you. With this, I'm going to finish here. I hope I made chromatin interesting for you, for those of you who don't work with chromatin. And I hope I transferred the message that mass spectrometers are really powerful machines. When you look at the masses, at delta masses, at labeled molecules, you can look at multidimensional data and dig deeper into what is happening into your system. We are particularly interested in this accessible heterochromatin domains because we work with conditions that could be potentially treated by targeting the proteome that occupies this red domains, right? So we can find, if we find inhibitors to prevent this from happening, we could potentially delay critical conditions. And then finally, you know, just as a last sentence, like I say, we just started working with the Saldivo system. We are now their United States training site. So those of you who are interested in that, you know, we are helping them train next generation of scientists that wants to work with these devices. And that's it. Thank you very much for listening, for being here. Thank you very much to the lab and all of these people and the funding. And thank you, of course, to Thiago for inviting me. And I hope this was interesting for you. And I guess I'll take questions now. So thank you again.
Thank you, Simone. Great talk. Yeah, I was sure that that would be great.
Thank you.
So I think we already have questions from the audience. So yes, Julia Cunha. She's asking a question regarding the histone post-translational modification turnover. Have you investigated that? Which one is more stable? It's a lot of questions. So
yeah,
which one?
I see that.
Okay, is stable. Have you observed more stability on old or new synthesized histones? And based on all MS data, on all combinatory histone PTMs, did you find a general functional whole for any histone PTM?
Yeah, that's a lot of very interesting questions.
Yes, and then if I can add, in the end, if you can just say that, do you believe the histone code exists?
No, don't make me say that. But first of all, the turnover, it looks like a detail, but it can really screw up your experiment. Protein modifications, they have different turnovers. A phosphorylation, those of you who do phosphoproteomics, you know it very well. Phosphorylation sometimes occur in milliseconds, like in the synapses of the brain. And histone acetylations, they might take, let's say, on average, an hour to be deposited on a protein. Histone methylation can take days. So this is not just a trivia. This is a very important thing to know when you plan an experiment. If you're studying an inhibitor of a methyltransferase on an enzyme that puts methylation, you put it in your cell culture, you wait one day and you go and look what happens. Probably nothing happens, because some modifications have much slower turnover than others, and this is important to know because time is essential in your biological system, and there are reasons why some modifications are faster than others. Many of them we don't know them yet, but for example, makes totally sense that phosphorylation has to be really quick, because phosphorylation regulates a lot of synaptic communication, and your brain has to think very quickly. But yeah, this is a very good question. And then which is more stable? I hope that answer it. Have you observed more stability old or new synthesized? Well, this is a difficult question. I can say this: studying new versus old histones goes a long way. Some data that I haven't shown you is that newly synthesized histones are very rarely unmodified. So why this is surprising? When you produce a new histone, like your ribosome is synthesizing a new protein, of course this protein is unmodified. But when we look at the abundance of histones that have zero modification, they are usually unlabeled when we do this labeling technique. What does it mean? It means that unmodified histones, they remain unmodified if they are hidden inside inaccessible heterochromatin. If they are accessible, very rapidly they get modified. I'm not sure if this answers the question, but what I mean is new histones, yes, of course, we don't have any number to prove that they are more unstable than the old one. But what is interesting is that new histones, even though they are synthesized unmodified, if they remain accessible, they get modified very rapidly. And the last question is, do you find that that a given histone PTM type, I guess, as a general function? And the answer is no, and that's why it's interesting and complicated. So we know, for example, that acetylations, they tend to be more associated with open chromatin. Part of it because acetyl-CoA is chemically reactive by nature. So even without enzyme, if a histone is accessible, gets partially acetylated. But we don't know exactly, we are not in the position yet now to say histone acetylation is a marker of transcription, histone methylation is a marker of gene repression. We know it, and you see it with all of this combinatorial code, that is more complicated than that.
And what about the histone code?
Oh, the histone code, yeah. So when I was a PhD student, I used to love to write histone code in a lot of occasions, presentations, and so on. And I've always been yelled at saying that if I say code, it means that it means that you have a meaning for every unit. What does it mean? A traffic light has a code. So the red light means stop, the green light means go, right? The genetic code has a code because we know that a specific triplet is always associated with a certain amino acid in translation. Not always, but let's say the majority of the time. To say that we have a histone code, it means answering the previous question. It means saying that a specific combinatorial histone modification is associated to a specific DNA readout. And we are not there. And therefore, when I say code, I'm a little bit cheating because we still don't have a meaning for the units that we observe. We have seen a lot of things, but as you can see how complicated the system is, we don't have the final answer. It's a fantastic hypothesis, and that, of course, this is moving our field very much forward. But most would argue that it's soon to be called a code.
Great.
So this was a politically correct answer.
I know. I put you in danger. Yes, I think Marcela has a question here.
Hi, Simone. It's
Hello.
so nice. Great. It's great. Huh? Sorry. Okay.
Let me just say something.
Sorry. I think everybody can turn on the video now. It would be nice to see all your face. And also, in the end, please keep here because we are going to try to take a picture in the end.
So actually, I am biased to talk about Simone because I am a big fan. So I think he did a fantastic work. So miss you. And I have actually a couple of questions in general. One, I got very interesting, this directed infusion that you mentioned, but I get concerned about the complexity because usually we do prefractionation to decrease the complexity of our sample. In this case of this directed infusion, you usually do a prefractionate offline and then some directed infusion to how can I say, get better of this complexity?
Yeah, I'll tell you the truth. We don't do that with proteomics. The reason because we do it with histones is because histone can be purified. So if you inject an entire proteome, it's really complicated. You get data that I would argue they're still not possible to fully deconvolute. There are some interesting papers on bioarchive. I think there is one from the laboratory of Steve Gigi that he does direct injection, and then he combines this with ion mobility, to get some sort of separation, and then there is some proteomics performed. And for sure it's a very good work, but I don't think we are in the place yet to perform proteomics without chromatography, just because of, it's not only for the number of peaks. Like I said, mass spectrometers are really fast, but they don't care if there is 100,000 peaks to select. The problem is of the dynamic range, right? And then you have species that are suppressing the others, and then you don't detect them. We do it with histones because histones are relatively simple to extract from all of the other proteins. And then something I didn't mention, but it's probably intuitive, histones, there is a lot in the cells. In the first slide, I showed that you have a nucleosome every 147 base pair of DNA. If you think about a 3 billion base pair, there are tens of millions of histones in every cell. You compare that with the abundance of certain enzymes, which are the ones that are always the most interesting for you. There are maybe 10 or 100. You're never going to see those enzymes in a full lysate.
Yes, thank you. And then about this that you told that you, let's say fish, you enrich for the stones. And then I also get curious because we know that usually these fishing pull down, they are never 100% efficient. And then it was just about how is the efficiency of this histone extraction, and then also the efficiency of what you mentioned about the separation of euchromatin from heterochromatin, and also from the quantification.
Yeah. This is a very good point. And this is always a problem that proteomic people have when they speak with people that don't have proteomic experience. Because people who don't have proteomic experience, they always ask you, can you tell me which proteins are in this tube? And we know that there is a lot of proteins in every tube. If you breathe into a tube, if you take water from the sink, you will find proteins, right? So that doesn't answer your question. So the way we approach this, I don't have an exact answer about how efficient is the histone extraction. It's still very high, so we're happy with that. But specifically about this fraction, you're totally right that you find lots of the same proteins in a lot of fractions. So the trick here is to look at enrichment. The same thing when you're looking at immunoprecipitation. What you want to know are the proteins binding to my base more enriched in my immunoprecipitation compared to a control? Because if you just look at which proteins are there, you're going to find the histones, you're going to find the ribosomes, you're going to find all of these proteins that are present in millions of copies in every cell. It doesn't matter how good your enrichment strategy is. So it's all about relative changes. It's never about getting the perfect purified protein. Now we're working here with, just to let you know, we're working here with a protein production facility, and they take pride of producing proteins that are ultra pure for cryo-EM, for X-ray, for all of these things, and then they gave it to us to characterize them, to prove that their protein is there. And we always find at least 100 proteins. So that's, it doesn't matter how well you do it, right? This is normal.
Yes, but then it is very reproducibility, this extraction, because if you have not some good reproducibility, you could have a misinterpretation of this quantitation too. So.
And that's the most important thing. That's why this direct injection, that's why we like it so much. Mm-hmm. It is less sensitive than chromatography, but it's more reproducible. And for lots of experiments, people don't want to know everything you see. People want to know, is my protein reproducible? Reproducibly more abundant in this system compared to the other. And you're surprised when you work with doctors, they don't care at all that you can detect 7,000 proteins. They just want to know, am I certain that this protein is more abundant here than there?
Thank you. And then I just have maybe more two questions, because I got very interesting also when you told about that chromatin, histone modification that you see in the open chromatin, but it's not correlated with the translational, yes? And then in this case, I didn't work with histone, so I got confused if in that histone, you have just that two modification, or you could have others modifications, how can I say, surround that position. Because maybe what is happening is a kind of an interplay of different modifications in different positions of this histone. And this is why it's not translating, and it's not correlating with the translation.
Yeah, that's true.
Transcription, but I didn't know if it was just one part of the histone or it was considered the whole.
Yeah, no, it's a good question, and that's because it was confusing because I didn't explain it very well. What we were looking at specifically there, we were looking at the typical bottom-up strategy. So we cut histones into short peptides. So we can tell about combinatorial modifications, but only if these modifications are on the same peptide, right? And obviously, this is only true for things that are very close to each other and things that, you know, where you don't have any cleavable site of your enzyme. So in that specific example, the reason because I only talked about K9, K14 is because these two modifications are on the same histone. But you're right, I don't know what else is on this histone. It's probably a mixture of a lot of other species. It's just that there was a clear example to show, but you're right, that is not the full picture.
Okay, thank you. And then one that I also got fascinated about this 3D extraction. And then in this 3D, I didn't understood correct, sorry, but it's kind of a 3D of only one type of cell, or you can have like a mix, because I know that there is the 3D of like mini brains and so on, and it's like a mix of different types of cells. The one that you are doing is a packet of the same cells, or you can have some different cells there?
Yeah, that's a very good question. So this is a very long story, but there are different ways of having three-dimensional culture. One is to have organoids, you know, to take pieces of tissue from real people and then maintain them in culture. It works, and of course they are highly functional because they just come off real people. They don't stay alive very long, at least in my experience. We do another approach. We start from a cell model, and then we just duplicate it until it forms something that can be maintained for a very long time. Now, what we do it only with one cell type, but you can mix in the same bioreactor. You can mix multiple cell types, and of course, people do it. There is one problem though. For example, liver. Liver is mostly made by 4 cell types. Now, don't make me say them all because I'm gonna look stupid, but there are these different cell types that are present in different abundances in the tissue. And when you put this in a cell culture, these different cell types, they might replicate at different speed than in a human. So you end up with a liver 3D structure that contains all of the cell types, but they are in completely different ratios than what you would see in a real liver. So yeah, you can mix multiple cell types, but it doesn't mean that they automatically become modeling a tissue better than just using one.
Yeah, and then just the last question about this 3D. So sorry, Hisham, I can see the Hisham again. It's just because since you can grow for a long time, I thought that maybe could be an interesting model, like to study the age, what is happening maybe in the age process and what is changing the chromatin. Is just maybe a curiosity or a comment.
That's a very good point, and that's one of the reasons why we really like that, because we study epigenetics, right? So epigenetics are these conserved traits that they're conserved in a long time. And to do that in a cell culture, you need to have a system that stays alive for a long time. So in one of the slides, you saw that we were studying some of these steroids that were 64 days old, so they were more than 2 months. We killed those because the coronavirus came to New York, but they were growing happy in that system, so we couldn't maintain them for longer. Now, you know, it can become unfeasible to do experiments that last forever, but so far we still don't have an end date. Potentially they can grow for much longer, maybe.
Nice. Thank you very much.
Thank you.
So thank you, Marcel and Simone. Now, Richard has a couple of questions.
Hello, Richard.
Hi, Simone. It's been a while.
Just a question, Richard. Is your couple of question 2, or it's like Marcel, a couple of questions with the end?
After Marcella, my couple is coupled to the end. I know, but well, very nice presentation. I see it's been a while. Now I see it's 6 years since I left Denmark. So
yeah. I have white hair in my beard.
But very beautiful work you've been doing. I was very, very impressed. Well, I was sure you were going to do great work. But anyway, so I have 2 or 3 questions, but they are more not specifically about what you presented, but it's just raised my curiosity to see if you are also tackling these issues in the field. So one of them is, given your expertise in studying histones modifications with mass spectrometry, have you tried to apply this to formalin-fixed tissues in order to correlate pathology and histone? And we have all these pathology banks with all these cheap samples. Is it possible? Is it quantitative, only qualitative? So
it's a very good question. I saw there are papers where they show that you can extract histones from FFPE, and the result seems to prove that the results are really, really similar. So I don't think it's a big deal. It's a lot of extra work to extract histones from FFPE compared to frozen or fresh cell pellet, and we still don't have. So the main problem of doing that, I don't think, is the extraction. I think is to find a good biological question, because you have these huge banks of cancer cells, and you're going back there and you analyze them, and you can detect that there are some changes in the global levels of histone marks. But people, my feeling is that people don't do it because they are still not sure what is this the answer to. Now, is this the answer that there is a mutation in a specific histone writer? We know that already because we have done DNA sequencing of these samples. And that's what I think is preventing people to do this kind of experiment. Now, can we stop the YouTube recording? I want to say something that is inappropriate, but it's okay. So there was a paper that came out last year in Nature where they did the histone modification profile in all the cancer cell line encyclopedia. And then basically it was more than 1,000 cell lines, different cancer models. And then we have now data about the relative abundance of histone modification in all of these cells. Who has used this data? You know, so far nobody. I'm not saying it's a bad study. I'm just saying it was a humongous effort. And what is the outcome? I know it's inappropriate what I'm saying, but because it was a very good work and I know very well the authors, but we are not even using those data because this is not quite the question we want to answer, like how abundant are histone modifications. That's why we are so interested in this other dimension.
Yeah, I understand. But at least you could have some data about correlation, maybe not causal, but correlation for sure. But anyways, and the second question, other questions I can make afterwards if nobody wants to make questions, which I'm sure it's not the case. I was curious about the clipped histones. So do they have biological significance, or is just a degradation? What's
it's a very controversial argument. We don't have a final answer. The papers that have been published about clipping until now, they show that if you have a clipped histone, the nucleosome is more unstable, which means that histone clipping could be a modification that is used by the cell to promote the eviction of the nucleosome. So it will be associated with active transcription. But then there are also examples of clipping that they cut histone tails that are hyperacetylated. So you remove the acetylations this way.
Yeah.
And what does it seems to mean the opposite. So there is not a final answer. Also because, as you can imagine, it is not that easy to discriminate clipping from just degradation of the protein. Because clipping is never super specific. Unfortunately, I don't have an answer to that. But the papers we have so far suggest that it's a mechanism of nucleosome eviction.
Okay. Thank you. I'll be
Thank you.
quiet now.
Thank you, Richard. Great questions. I think now Deborah has a question, right?
Yeah, can you hear me?
Yes.
Yes.
Hello, hi,
Hello.
greetings from Sweden. So I think Marcela asked most of the questions that I wanted to ask about mass spectrometry. But if no one else has, I don't know, I was going to ask you to talk a little bit more about how to apply and what are the problems you saw in using SciLac to study the changes. Yeah, I'm interested in that. So maybe you could talk a little bit more about that.
Yeah, it's a good point as well. I try to not go into the technical details in this presentation because it was already heavy enough. But it is fascinating for us as we develop methods. Right? If we want to label a protein, how do we label it? And it depends also how you process your samples. For example, when we want to study this histone turnover, we analyze our short peptide. We have a sample preparation that makes sure that every peptide contains one arginine residue. So we grow the cells into heavy arginine media because then we have only two signals, the light and the heavy. If you have a peptide that has multiple sites that can be modified, then you have this smear of isotopes and becomes more difficult to quantitate. Another important thing is that whatever amino acid you pick has to be essential, has to be used by the cell directly, is not produced internally. So when we look at histone tails, they have a lot of arginine. We cannot use arginine. But what is interesting, for example, on the tail of histone H3, there is only one histidine. So we use heavy histidine if we want to analyze the turnover of histones, analyzing that with middle down. Because the amino acid needs to be essential, and it's much more convenient if it's present in a single copy in the protein sequence. Then how long do we keep it? The interesting thing, so compared to the classical SciLac, where you have your light culture and your heavy culture, your heavy culture needs to be completely heavy, right? Otherwise, when you mix them, you have, you don't know if the protein is coming from the second culture. In our case, it's much, much easier because we don't care about complete labeling. We just care about how much a certain histone is more labeled compared to another. So if the labeling is 30% or 60%, we don't care. So when we do this culture, we don't even prepare special media. We just take our normal media and we just put inside a bit of heavy amino acid. And we know the cells will eat it.
Okay. Interesting. Thank you.
No problem.
Okay, great. Now, Davidson, ask it for a question. Then it's Ana Gisele.
Could you hear me?
Hello.
Hello. Congratulations. Your work is very interesting.
Thank you.
I've been working more recently with a chromatin organization as well, but using like imaging techniques such as super resolution microscopy. And these days there is a hypothesis that is related to where are the most active zones in the nucleus. So these days they are saying that the most active zone is going to be more in the middle of the nucleus, and the less active zone is going to be close to the edge, so close to the nuclear envelope. So I was like interested to know if you are seeing like if you can correlate your data with the binding maybe of histones with the lamins that's supposed to be the less active zones.
Wow, I could speak for hours here. This is very exciting. I really like super resolution microscopy. Unfortunately, we don't have it here. So it's very cool technology. Regarding the position in the nucleus of the active chromatin, this is very interesting because like you saw in my first cartoons, heterochromatin is defined to be associated with the lamin, which is a protein that decorates the surface of the nucleus. So up there, you have a lot of silent chromatin. So you would think in the middle of the nucleus is where you have the active transcription. But the most active transcription, you have it for the chromatin that is next to the nuclear pores. And this makes a lot of sense, right? Because it's very easy to send RNA in and out. So nuclear pores are also at the nuclear periphery. So yes, it's true that in general, heterochromatic domains are on the borders of the nucleus, and there is more transcription overall in the center. But we have a lot of exceptions. So I will be careful in saying, the chromatin is here and therefore is active. But super resolution microscopy really goes a long way because you can really see with this high resolution how compact certain chromatin domains are. So I actually believe that this technology will be really, really helpful to answer some of the questions that we cannot answer with mass spec because we extract both histones. So we don't know anymore where they are. It's a very cool technique, but I'll be careful in saying the chromatin is here, therefore is active.
Thank you. Yeah, I believe exactly what you said. Every technique has the constraints, and I have been working a lot with real-time imaging as well. These days, they are showing that some nuclear proteins and nuclear pore proteins could have other like functions. And these days, actually, I'm very interested how to correlate like mass spec data and real-time imaging data and mass spec data. Maybe in some point we can collaborate to answer new questions in this field.
Yeah, no, we don't have that technology, so we really like it. We would really like to discuss that.
Thank you.
Great, Davidson. Ana Gisele?
Thank you, Thiago. Congratulations for the organization of this series of seminars, and congratulations, Simone, for great talk. Sorry for my neighbor that is rebuilding now. He's slow. I just would like you to comment a little bit on, well, we are protein lovers, right? So we tend to forget a little bit about the others partners in this scenario. So could you comment a little bit on DNA modification and how complicate even more your histone code or whatever, if you have a code or not, but this complex interplay of codes that maybe or maybe not will have a different output, for example, depending on the decoration that you have on the DNA segment that is around the histones that you are looking at.
Yeah, you got me. I tried to twist the presentation to show that histones do everything, but you got me. It's true. DNA methylation, for example, some scientists believe is the only real epigenetic mark, and it makes sense, right? Because when you produce new DNA, if you have DNA that is methylated and you get, one of the strain goes to one cell, the other one goes to the other cell, the methylation of the DNA is the only thing that survives to the process. So that is the one inherited in the new cell, and then histone modification comes afterward. So DNA methylation is very important, and it's arguably the driver of many of the histone PTM deposition. Mostly, so the lysine 9 trimethyl that I mentioned, that is one of the first recruited. So when regions of DNA that have DNA methylation, they tend to be decorated with K9 trimethyl. And in fact, if you knock, and we have data for that, if you knock out the 3 DNA methyltransferases, then you lose K9 trimethyl as well on the histone. And this will be a long story. The thing about heterochromatin is that there are multiple types. So lysine 9 trimethyl and DNA methylation, except in stem cells that are in the developing stage, but in adult cells, most of the time, they don't silence genes. Genes are only 2% of the DNA, and there is a lot of DNA methylation, there is a lot of K9 trimethylation. Most of this DNA is actually in charge to silence other types of DNA, transposons, satellite repeats, ALU, sometimes even like dead viruses that have been dead on our chromatin for 1,000 years, and we still didn't get rid of it. So this DNA methylation plays a critical role in structurally silencing heterochromatin that we don't want these cells to express. And that's why when we develop cancer and when we age, and when some of this chromatin becomes accessible, that's when we have these retrotransposons reactivating and the cells get sick. It's not the only thing that explains aging, because aging is a much more complex biology. But you're right. You know, we get DNA hypomethylation in cancer. We get DNA hypomethylation in aging, and that's when these things happen. And that's you know, the data that we have show that this is very detrimental for the cell. So yeah, of course, DNA methylation plays a critical role in regulating histone modification, in regulating chromatin state, and there are many modifications that we haven't characterized yet. We know a few DNA modifications. We know more than 100 RNA modifications. And you know, I was reading a paper a few weeks ago about RNA acetylation. There's still so much we don't know about these modifications. Part of it because we don't have the right technology. Because when you do sequencing, you eliminate these modifications. So they're not easy to map.
Great. Okay, thank you.
Thank you. Oh, Tiago, I think you're muted.
Of course. That's a wise thing to do, right? Before asking something. So is there anyone that wants to comment or make a question? Otherwise, I have one quick question. No one? No? Anyone? Okay. So I have like a really, it was really important to me and kind of scared me out. And I know that probably for Hisha as well, Marcela, and who else? Carol and Felipe, all the people that's like hard drinkers. Who else? Juliana Cassoli, which is hiding there, but I know that she also likes a lot of bull. So just think about the alcohol and how it changed the isotope pattern of the histone modification, right? Does it, is this modification spread through the generations? Like after you make like the insult with alcohol and then it changes the isotope pattern, and what about if, when you take it out, is it like, does it continue to have the same pattern?
That's a good question. So that's how we sell it for now, that histone modifications are epigenetically inherited. And in fact, when this paper came out, the group leader that published this paper, she went on TV, she was on Fox News, which, as you know, is the favorite channel of Donald Trump. And of course, what they were discussing about is that is this modification impairing memory? Does this explain why when we drink too much, we have a neurodegeneration? And the answer is I think is more complicated than this, because like I said before, histone acetylation has a turnover of about an hour. It goes away. It gets recycled very quickly. The question is, once it gets exchanged, is this chromatin remaining in the same state? And there's a lot of extra regulatory mechanisms that we don't know. So to answer your question, no, that molecule of ethanol is not inherited through generation, but is this creating long-term changes in the chromatin state? That could be, and that experiment didn't prove it, but yes, it's an excellent question that we don't have a definitive answer for. Yeah.
Great. Thank you a lot. I think Marcela has a question or no? Yes?
No, Thiago, remember, ignorance is blessing sometimes. Stop asking those questions, please.
So keep drinking.
Good. It's just because about this alcohol, and then I also remember because the people are seeing that exercise or even the microbiota affects future generation, and then this could be also something correlated to these histones modification or sensibility that probably is modified. Yes. Oh.
That's a very good point, because acetylation is the perfect example. Acetylation comes from acetate in little part, comes from glucose in some part, and it comes from butyrate. And who processes butyrate in our body is the microbiome, is not us. So yes, if you change the microbiome in your gut, in your intestine, you change the histone patterns of your cells, because many of the nutrients we get them from them, and there's a lot of very cool science on that.
Yeah. I was going to comment. That's like my final comment, and then we are ready to go to us for lunch. I don't know about you, Simone.
I'm 1 hour earlier.
Yeah, it's lunchtime here. It's just because like Ana Carolina, which is also attending here, she's a PhD student in our group. And I want to later make a meeting with you because she's going to study microbiota. And yeah, I think you could help us a lot.
Well, thank you.
Yes,
I wanted to say before I forget, I lost my opportunity because she just disconnected. But something you might want to know that the Sidoli lab stands on the contribution of a very important Brazilian postdoc.
Yes.
I'm sure some of you know it already, but everything you've seen, there's a big Brazil slice in all of this. So yeah. I thought you wanted to know.
Yeah, sure. But we are going to talk later if you want. Yeah. Okay. So I would like to thank you again, everybody that lasted here until the end, and remind you that next week we are going to have another talk Wednesday with Fight. Some of you already know him. He's a German guy who speaks Portuguese, but I think it's better to ask him to speak in English because I think you're going to understand better, isn't it, Risha? Yeah, I think it's better that he speaks in English, right? Yeah, but bring friends and thank you again. See you next Wednesday.
Thank you, everybody.
Bye-bye.